So my little finger here, is this a mutation or is this just part of the variation within our species? Physical difference that you can compare within what we would call an invariant. Now, if I'm standing right here and all of a sudden I start sprouting horns, or maybe I get some wings so I can fly, that'd be cool. There's my DNA. This would be something that would be unique to me, and it's going to disappear when I disappear, when I cease to function. All right. Maybe I'll look darker eyebrows or some fangs. That doesn't look like fangs. But anyways, what exactly is that? Mutation. What causes that? And it's actually the mutation in the DNA. Brought to you by Curious Marineland. Now here, the long-term impact would have to be tracing those changes if they get to the reproductive cells and pass on through generations. And I'm not going to go into all the details now. I'll do that in a later video. So pretty much, I want to talk about mutations. What exactly is it? It's a change in the DNA. It's a difference from the source. All right, the source we often call the wild type. Now, in class, I do this exercise. I put this up here and say, hey, what's causing these deformities? These are all somatic cells. And today, frogs, amphibians are the modern-day canary in the mine shaft. They're a warning beacon that there's something wrong with the water. And if you pay attention to the news, we really need to have clean water because it affects their development. See, they develop externally, so the pollutants can diffuse through their membranes of the developing egg sacs, and that's going to affect their body cells as they develop. So that's what it is. So here's your basic definition. This is something that my students are expected to know. It's a change in the DNA sequence. What the impact of the change in the DNA sequence is what we'll do as we go through this video. I'm going to do a couple other videos that go into this. So this is my series on mutations. So is it harmful or have no effect? And it can also be beneficial. So I want to just identify the types, and I'm going to show you some elaborations in part 2 of this. So a gene sequence of DNA. And all right, so change in sequence, the deformed proteins. Now this can cause problems. When I say deformed, the change in the DNA changes the RNA, which in turn can change the amino acid. One amino acid out of place can be harmful. One amino acid changed could have no effect at all. Now let's say it's a body cell that controls the gene regulation or the mitosis. That's uncontrolled old mitosis. That's what leads to cancer. So here's your chromosome. So we got to review. DNA, chromosomes, genes. All right, so DNA is the 4 letters A, T, G, C. A certain arrangement, a specific order or basis is the code, and that's what makes up a gene, and a bunch of genes strung together makes up a chromosome. And so you see this whole thing is a chromosome, and each one of these bands is a segment of a chromosome that has a specific order of bases that determine traits, meaning they determine the RNA that will be made, and in turn the translation will happen to make those proteins. Give you a wider look here. This is this is the first base and this is the last base. So there's a whole bunch. This is a huge gene sequence, and you can see at various points if there's changes here, that's what the mutation is. Now, again, we're not talking about variation in species. We're just talking about change in mutation. Variation in species is the subtle differences between one person at this location versus another. We actually call it at this locus. All right, so here there are 2 types of mutations. So I'm not going to do much longer with this video. I'm going to expand. I'm going to do a different video on chromosomal ones and point mutations and frameshift and all these. So these are the 2 ones that my students see first. So let's talk about gene mutation. All right, point is a change. Now we're on the gene level, not the chromosome level. Chromosome is multiple genes stuck together in one area, and that we call a chromosome, or the chromatin that condenses to become a chromosome during mitosis. There's 3 examples that I'm going to show you. There's actually a 4th, but there's 3 that are easy to see: substitution, deletion, and insertion. Now 2 of these cause what's called a frameshift, where the letters move down. You delete one and all the letters rearrange, or you add one and all the letters rearrange. And these 2 can have the biggest impact on the results. Versus substitution, it depends on which base. Remember, every 3 letters is called a triplet, and every 3 letters of mRNA is called a codon. It depends on which base in the 3 letters. If it's the 4th letter, you might not have as big a change as you would if it's the 1st letter. So just a reminder, there's genes. So this is the DNA, this is the RNA, this is the amino acid. We're just using geometric shapes for this. So notice the difference. Right? Here's the DNA. This letter is out of place, and if you notice, this is the three letters, 3 letters, 3 letters. All right? So, all right, let's go back. We're back. So 3 letters, this middle one. This is the messenger RNA. Notice what this one has changed. That means that letter's going to change. And notice the shape. Here's the difference. Now, this is geometric shapes to show, but if this is the part of the folding protein, this one letter place could change the shape of this function of this protein, and hence change the function. It could enhance it, or it could make it worse. Okay, so let's do this. To show you an example of a substitution, here we have a phrase: the fat cat ate the rat. And I need to change the pen here. All right, so let's go back. I'm back. Okay, I'll put me over here. Now, again, that first one is the phrase, the fat cat ate the rat. So a substitution, if you notice, when we change the F and the R, now it's the rat cat ate the rat. It still makes a statement. It's not, you don't lose some of it. Now, let's do deletion. The fat cat ate the rat. Now, if we take off that T, the fat cat ate the Ra, like the sun god or something. Or, watch this. We delete this E right here. And then this is an example of a frame shift. All right, see if you can follow the letters. There's the fat cat, so if we get rid of this E, all this moves in this direction. All right, all this moves in this direction. All right, now this teeth, or maybe it's Klingon or something. You actually lose the phrase. Imagine this being the protein that you lose. Okay, addition, same result. The fat cat ate the rat. Now, if we add a base to the first word, so you see if you can follow this. I'm going to come back to my pen here. Here's the original. Now we added something at the end. Now it is shifted. So that's, again, that's what your frame shift is going to do. And if we go back to, so I have some other examples. Let's look at the actual DNA here and see. So let's follow this example. Okay. So take a second. You got here's the GA, and then there's a corresponding complementary strand, and let's watch what happens. So this is our template. In genetics, we'd call that our wild type. So imagine this is what a parent, and this is what they send to their children. So in that case, it is a change in the DNA that gets passed on to the next generation. All right, so what's the first one? That is a substitution. Okay, let's do another example. What is this one? Here's your original. Here's the difference. That is a deletion. All right, that's going to affect the amino acid that's produced. All right, so here's a third example. That is an insertion or an addition. I'm going to give you a third one that wasn't in the notes earlier. And this, see if you can see what's happening. This is an example of an inversion. Right, and just to follow, just take a look at how they would affect a sentence. Now, this is not a sentence in your body. This is, imagine, a sequence of amino acids. Okay, chromosome mutations. So I'm going to go and close with these examples, and then I'm going to do another video that elaborates on chromosomes, and I'm going to show you some actual areas where you see the DNA and it changes. So let's just go ahead and take a look at these. Now, what is the difference between a chromosome and a gene? This is a whole segment of genes. Imagine each one of these being like 100 nucleotides or 1,000 nucleotides long. That has a greater impact. And this one translocation is very important to understand that one, because that can lead to Down syndrome and some other ones that I'm going to talk about. So deletion, look what happens. It's the whole piece. Duplication, this is how we actually get what's called increase in information. And evolutionary-wise, this is something that allowed for chromosome numbers to change or increase, and then it increases, and then those changes cause variants that end up being favorable or unfavorable, depends. The favorable ones are here because they've survived and reproduced as part of natural selection. Okay, inversion. And then translocation, a piece of the chromosome that breaks off and attaches to another. See, these are different genes. These other ones were genes were kind of relocated or shifted in another direction. When you shift, like inversion, that may not infect gene. That may not have an impact on the gene expression. Okay, so we've talked about them being harmful. Let me show you some other examples. This video is going on way too long. So this is the famous Kittering bird experiment with industrial melanism. What you didn't see was this little critter right here survived because the bird went after that one. This survived and reproduced. Okay, sickle cell is another example. Let me show you. It literally is one letter on the DNA that gets transcribed, that gets changed, and then it is transcribed, and you get valine where you're supposed to have glutamic acid, or glutamine. That's glutamine. And that causes the sickle cells, which is going to, the impact is going to cause anemia, and your blood cells can't make it through the blood vessels as well as they could because of their sickled shape. And so cystic fibrosis is another example. So just to show you some other examples, just to kind of stop the video with this, these are the types of mutations that we study, and there's various disorders that we look at, and we're going to, when we get to genetics, we're going to go into this in greater detail. So that's pretty much that's it. Brought to you by Curious Moranland.